today I will be talking about DNA organization. I have told you that the genetic information which is present in the DNA of a molecule, that is the sequences in which the bases and nucleotides are arranged. That is the sequence of the DNA, and that is the genetic information. And every three nucleotides, they form a codon, and one codon codes for one amino acid, and the peptide chain is formed by reading that code, that is called the genetic code. That is the sequence of the amino acids or the sequence of the nucleotides arranged on the DNA. So this genetic information in the DNA of a chromosome can be transmitted by exactly replicating it. DNA has to be replicated, another cell has to be produced, and exact same sequence of DNA is given to the offspring. ठीक है, ऐसा नहीं है कि एक डीएनए में सीक्वेंस कुछ है और अगले डीएनए में सीक्वेंस कुछ और हो जाएगी. Same sequence will be carried to the next generation, or it can be, at times it is exchanged by a number of processes, which is called crossing over or recombination or transposition or conversion. So us case mein the sequence will be changed and that will be inherited. So that sequence is either same or it is exchanged, but this exchange is also very planned. And किन किन conditions में होता है वो हम बाद में हम पढ़ेंगे. Chromatin is the chromosomal material in the nuclei of the cells of eukaryotic organisms. As I told you in my previous lecture, that in prokaryotes, the chromosomes are circular, present as circular molecules, and they are not in the nucleus. They are present in the cytoplasm as a nucleoid. But in the eukaryotes, there is a proper nuclear membrane around that nucleoid material, and it becomes a nucleus. So chromatin in the eukaryotes is present as chromosomal material in the nucleus of the cells. So 158 have joined, so I am starting. So good afternoon again. Good afternoon, everybody. Now I think sufficient people have joined, so I will start with my slides. And if I remember, I'll stop after 15-20 minutes and look after the questions if you have any. Okay? Otherwise, I'll take up all the questions at the end of the session. So DNA is organized into chromosomes, and this property we call as DNA packing. How do you fit all the DNA into a nucleus of the cell? So what happens to the DNA, linear thread like DNA को क्या हो जाएगा? It will start coiling and folding. So DNA के अंदर आपको, you have to remember these terms, that it is a double helix molecule. It is arranged, coiled and folded to form nucleosomes. There is a thin chromatin fiber. There are loop domains, and then a full chromosome will form. I'll explain you all these things in further slides. See, the organization of DNA, human diploid genome, which is 7 into 10 to the power 9 base pairs distributed over 46 chromosomes. There are 23 pairs of chromosomes, which we discussed yesterday also. Of these, 22 are autosomal and one is 23rd chromosome. Pair of chromosome is the sex chromosome. So the human diploid genome will have 7 into 10 to the power 9 base pairs. इतने nucleotides हैं total हमारे सारे chromosomes के अंदर. And when it is placed end to end, अगर हम इस पूरे chromosome को खोलें तो the length will be around 2 meters. If one nucleotide is added per second, then it will take around 250 years to synthesize whole DNA of the human cells. So see how all these processes are so well coordinated and so fast. Otherwise, अगर एक सेकंड में हम एक न्यूक्लियोटाइड ऐड करें तो 250 साल लग जाएंगे एक डीएनए मॉलिक्यूल बनाने में. and length of the DNA is compressed to 10,000 fold to generate chromosomes. Bacterial DNA is the closed circular one, which we have already discussed. Now, when we talk about the level of structural organization of DNA, so there are different levels of organization. So at the primary level, you see there is only the sequence of nucleotides, which is arranged from 5' to 3' direction with the help of phosphodiester bond. ठीक है, जो आपस में सीक्वेंस है वो फॉस्फोडाइस्टर बॉन्ड से जुड़ी हुई है, ठीक है. जो उसके बीच के हाइड्रोजन बॉन्ड्स है, दैट इज नॉट मेंटेनिंग द सीक्वेंस, दैट इज मेंटेनिंग द टू हेलिसेस. So the sequence of nucleotides from 5' to 3' direction is maintaining the primary structure with the help of phosphodiester bonds. Then this sequence, nucleotide का जो ये sequence बना, उसने turn लेना शुरू किया and it turns into double helix. So this becomes the secondary structure, which is taken care of by the stabilizing forces like hydrogen bond and the stacking of bases with the help of hydrophobic interactions. So next what happens? The tertiary structure comes by the supercoiling of the DNA, which we did yesterday. This is a strain to the molecule. It is ATP utilizing molecule and procedure, and it gives strain to the molecule. Okay, so this is taken care of by the DNA and protein interactions. And finally, the quaternary structure is maintained by the chromatin. Chromatin में क्या होता है? DNA protein interactions होती है इसमें भी, and there is a tremendous amount of folding and binding around the molecule. And how is this taking place? This I will tell you in detail. Before that, I will tell you some basics about the chromosomes. In humans, there are 23 pairs of chromosomes which are numbered according to their length and position of the centromere. So can you see in this figure that this center dot, this rust orange dot, can you hear me now? Am I audible now? Okay, got it. I don't know what happened. Anyways. So G banding or the Giemsa banding is a technique used in the cytogenetics to produce a visible karyotype. What is karyotype? Karyotype is the chromosome, जो हम chromosome का picture image देखते हैं, that is the karyotyping, and इसमें हम chromosomes को stain करते हैं Giemsa stain से. So that is the technique to produce visible karyotype by staining the condensed chromosomes. The pattern of bands are numbered on each arm of the chromosome from the centromere to telomere. I will show you the G bands in the next image. So in this image, you can see these chromosomes. There are bands in between all the chromosomes. These are the G bands. So ये जो G bands हैं, ये Giemsa stain से वो color लेता है, और इनके according हम chromosomes को identify करते हैं कि कौन से chromosome में कितने bands हैं और वो कौन सा chromosome है. ठीक है? So this is how the karyotyping is done. In the somatic cells, the genome is diploid, and each human somatic cell will contain 22 pairs of homologous autosomal chromosomes and two sex chromosomes, that is XX in female and XY in male. Okay? So you have to understand that somatic cells में genome is diploid and 22 pairs of autosomal chromosome and one pair of sex chromosome is present in the somatic cells. Whereas in the gametes, that is ova and sperm, only haploid genome is present. 22 pair nahi, only 22 autosomal chromosome and one sex hormone, either X or Y, as depending upon the gender, these will be present. Chromosome 1 is the biggest chromosome and chromosome 21 is the smallest. So what happens during metaphase when the cell multiplies? During the metaphase, DNA can be seen under the microscope as super packed chromosome where identical sister chromatids are connected at the centromere. And the centromere is, what is centromere? Centromere is the site for attachment of proteins that link the chromosome to mitotic spindle. The chromosomes, and the centromere region, it is rich in adenine and thymine base pairs. तो 80 rich region जो होता है वो centromere है. Metazoan centromeres are bound by nucleosomes containing histone 3 variant protein and other specific centromere binding proteins. So there are certain proteins which will be bound on the centromere, and this complex with the bound proteins in the center of the chromosomes is called as kinetochore, which provides an anchor for the mitotic spindle. So mitotic spindle attaches to the centromere and then it pulls the one arm of the chromosome towards the other side end of the cell where when the replication of the cell is taking place. So you should remember that what is centromere? It is rich in 80 bases, kinetochore, and so you have seen the centromere. Now I will tell you about another part of the chromosome that is present in the ends of the chromosome, that is the telomere. The ends of each chromosome contain structures called telomeres. The telomere consists of short TG-rich repeats, and the human telomere has variable number of repeats of the sequences. This is a particular sequence which is present in the telomeres, 5' and 3' direction में TTA, This can extend for several more bases. Telomerase is an enzyme which is a multi-subunit RNA containing complex related to the viral RNA dependent DNA polymerases, which are the reverse transcriptases, and this enzyme is responsible for the formation of telomeres. Telomeres में क्या होता है? As the cell multiplies, the telomeres keep on getting shortened, and अगर telomerase enzyme telomere ends of the chromosomes को synthesize करके chromosome की length maintain ना करे, so with every replication, after few replications, this chromosome will keep on getting shorter, and it will become too short to be viable. So it will die. Okay, it will be not stable molecule. So telomere shortening has been associated with malignant transformation and aging, and telomerase has become an attractive target for cancer chemotherapy and drug development. Each sister chromatid contains one double-stranded DNA molecule, and during interphase, the packing of DNA molecule is less dense than the condensed chromosome during the metaphase. So metaphase chromosomes are nearly completely transcriptionally inactive. So first I have told you about telomeres, that these are the, again I will repeat, telomeres are the structures of the DNA sequences which are present at the ends of the chromosomes, which maintain help in maintaining the length of the chromosomes. Otherwise, with every replication, the size of chromosome reduces. So they are there to stabilize the whole chromosome molecule. The ends of chromosomal DNA are not replicated exactly by the DNA polymerase. For this, I told you there is another enzyme called as reverse transcriptase, which is there to, you will see when you study replication, only then you will be understanding how is this taking place. But you just to make you understand the term telomere, these are the parts of the DNA present at the end of the chromosome. Shortening of chromosome is prevented by adding addition of the telomeric sequences by telomerase enzyme, and there is a 3' overhang on the 3' terminal end. और इसकी स्पेसिफिक सीक्वेंस है 5' to 3' डायरेक्शन में TTAGGG, और ये सीक्वेंस बहुत सारी चलती जाती है आगे. दिस सीक्वेंस कंसिस्ट ऑफ मेनी टैंडम रिपीट्स, एंड इसका कॉम्प्लीमेंट्री स्ट्रैंड में भी कॉम्प्लीमेंट्री सीक्वेंस होगी. So this is how a chromosome looks like. Center में you can see the AT rich region is the centromere. There are two sister chromatid. You can see one side is chromatid 1 and the other side is chromatid 2. And on the ends are the telomeres, which have TTAGG sequences in tandem. Now the chromosomal material is extracted from the nuclei of the eukaryotic cells, and it consists of very long double-stranded DNA molecule which can be around 1.7 meters long. Proteins involved in folding and packing of the eukaryotic chromosomes consist of histone and non-histone proteins. Histone proteins के details I will share with you. These are the basic proteins, and non-histone proteins are the other proteins which are associated with the folding of chromosomes. That is the regulatory proteins which include the enzymes and the other regulatory factors of transcription. Because DNA ने आगे क्या करना है, ट्रांसक्रिप्शन करनी, एक तो ट्रांसक्रिप्शन करनी है यानी कि आरएनए बनाना है, या रेप्लिकेशन करनी है या अपने आप को रेप्लिकेट करना है. तो इसके जो एंजाइम्स हैं वो डीएनए के साथ हमेशा रहने हैं. So these are the enzymes which are non-histone proteins. These are regulatory proteins which are the regulatory factors of transcription. ठीक है. और हिस्टोन प्रोटीन्स जो वैसी पैकेजिंग के लिए चाहिए होते हैं उसको मेंटेन करने के लिए पैकेज स्ट्रक्चर फॉर्म में. Also some amount of RNA is present in the chromatin. If the total DNA in one person was laid in a straight line, then it would stretch to sun and back over to 600 times. So if you know that the sun from Earth is 93 million miles away, so you can see the total DNA of the person is so long. अगर आप उसे stretch करें, that is all compacted in our cells. And if you could fit 1 million threads of DNA across the period at the end of this sentence. Now chromatin is of two types, euchromatin and heterochromatin. Euchromatin is the one which is transcriptionally active. जो chromosomes का DNA RNA बना रहा है, ट्रांसक्रिप्शन कर रहा है, एक्टिव है, उसके एंजाइम्स भी एक्टिव हैं, और वो डीएनए डाइजेशन के लिए भी सेंसिटिव है. उसको डीएनए एंजाइम कैन कट दिस दैट क्रोमोसोम, एंड उसके अंदर जी बाइंडिंग अगर आप देखोगे तो वो लाइट स्टेन लेगा क्योंकि वो एक्टिव है इस समय. तो ये euchromatin है. Similarly, those parts of the chromosome which are inactive transcriptionally, darkly stained, and are histone deacetylated. I will tell you what does this acetylation and deacetylation means. And they are sensitive, resistant to the DNA's digestion. They are heterochromatin. Okay? So you have to remember that chromatin, that is the chromosome material with histones and non-histone proteins, can be euchromatin, that can be transcriptionally active, or heterochromatin form. So the levels of packaging which we have already discussed, primary, secondary, tertiary and quaternary structure. Now I will explain in detail with the figures. First is the sequence of the DNA. Then that sequence takes a fold, and then it takes that takes us to those take supercoils, and then they start taking folds, and then they take form their loops and domains, and they finally become compacted into a chromosome. So naked duplex DNA, which is present as beads on a string created by the formation of nucleolus, and then it is more compressed into 30 nanometer solenoid, which extends to form a chromosome, and then gets more condensed and forms a mitotic chromosome. This 1 to 2 nanometer double-stranded molecule keeps on organizing itself in around a central scaffold. In this figure, I would just want to show you the telomere and the centromere and the heterochromatin and the euchromatin region. Orange color region is the heterochromatin, and the blue colored region shown here is the euchromatin. Centromere is the center, and the telomeres are the side ones. Chromatin contains DNA and protein in equal mass in some RNA, which I've already told you. Chromosomes contain 2 chromatins. Chromatin has 1 DNA and histone protein plus non-histone protein and plus some amount of RNA. Histone proteins are the basic proteins. You have to remember that they are positively charged basic proteins, and they are present as H1, H2A, H2B, H3 and H4. And then these 4, not 4 in the sense कि H1, 2, 3, 4 तो 4 है, लेकिन 2 के 2 फॉर्म्स होते हैं 2A and 2B. They form histone octamer. H3 and H4 का एक डायमर बनता है, और H2A और H2B का एक डायमर बनता है, और ये टोटल 8 हो जाते हैं, and they form an octamer. So this kind of octamer is present in the disc-like form, and around this is wound the DNA. Okay, I will explain all this in detail. Till then, if any questions, by everything is clear, you are all on with me. Any doubts? You want me to repeat anything? You can write in the chat box. Okay, then I will move further. And the H1 molecule is loosely bound to this histone octamer structure. So now we will do the details of histones. There are 4 classes of histones: H1, H2, H3 and H4, and 2 is again 2A and 2B form. These small proteins are positively charged at the physiological pH as a result of their high content of lysine and arginine. So why are histones basic? Because they are rich in lysine and arginine, and that is why they are positively charged, and they form ionic bonds with negatively charged phosphate group in the DNA. So why histone binds to DNA so tightly? और डीएनए क्यों हिस्टोन्स के अराउंड अच्छे से बाइंड करके और अच्छे से स्टेबल होके रह सकता है? Because DNA is a negatively charged molecule. Why is DNA negatively charged? Because it has lots of phosphate molecules. Phosphate is negatively charged, and this DNA, negatively charged DNA, is wound around and bound tightly to the positively charged histone molecules, and the positive charge of histone is because of lysine and arginine. As you can see in this figure, these blue balls are the octamer core of histones H2A, H2B, H3 and H4. Each one is in pair 2. And इसके अराउंड में ये DNA है एक ribbon kind of thing, red and purple, which is that it is wound around the histone proteins. And इसको बीच में ये green oval shape kind of histone 1. अभी तक हमने H2A, H2B, H3 और H4 के octamers की बात की है. H1 अभी तक हमने बात नहीं की थी. So H1 is the which links, attaches itself to the DNA from outside and stabilizes the molecule. So structure of all the 4 core histones that form the nucleosome have been highly conserved between the species. And the conservation of histone implies that they have the same function in different species and the entire molecule is involved in that function. Okay? So this extreme conservation implies that the function of histones is identical in all eukaryotes. The carboxy terminal, that is 2/3 of the histone molecules are carboxy terminal ki side se 2/3 of the histone molecule is hydrophobic, while at the amino terminal it is basic. 4 core histone molecules are histones are subjected to at least 6 types of covalent modifications. अब ये histones जो है वो proteins हैं, ठीक है? They have protein मतलब they have the amino acid sequences with them. इनको हम modify कर सकते हैं. अब एक DNA, just listen to me. एक DNA molecule which is wound around the histone proteins. अब अगर हम उस हिस्टोन प्रोटीन को केमिकली चेंज कर दें तो ऑब्वियसली इट विल ब्रिंग सम चेंज ऑन दैट डीएनए आल्सो. उसकी बाइंडिंग के साथ उसकी कोई वो हो जाएगी, कुछ तो वेरी करेगा, उसकी बाइंडिंग विल बी चेंज. सो H3 and H4 they form a tetramer containing 2 molecules of H3H4. ठीक है, पहले तो H3H4 का जो गया. H2 and H2B they form another dimer. Under physiological conditions, these histone oligomers associate to form histone octamer where the composition H3H4 के 2 और H2H2B के 2. ठीक है? So you can see here कि H3H4, H3H4, H2, H2B, H2A, H2B and H2A, H2B. This is how this octamer is formed. And इसके around this DNA is wound. इस इतना अच्छे से वो उसको अपने पॉजिटिव और नेगेटिव चार्ज की तरह से डीएनए को बाइंड अपने आसपास लपेट लेता है कि डीएनए की कॉम्पैक्ट होना स्टार्ट हो गया. ठीक है, दिस इज द फर्स्ट स्टेप. H2A and B they are lysine rich, and 3 and 4 are arginine rich. Histone synthesis stops when DNA synthesis stops. जितना डीएनए उतना ही हिस्टोन है. ये भी होता है, जितना डीएनए उतना ही हिस्टोन है. केमिकल मॉडिफिकेशन, जो 6 टाइप्स ऑफ केमिकल मॉडिफिकेशंस हो सकती हैं हिस्टोन मॉलिक्यूल के अंदर, दे आर एसिटाइलेशन, मिथाइलेशन, एडीपी राइबोसाइलेशन, फॉस्फोराइलेशन, सुमोइलेशन. ठीक है, एसिटाइलेशन एंड मिथाइलेशन ऑफ लाइसिन रेसिड्यूज इज द मोस्ट कॉमन मॉडिफिकेशन. तो हिस्टोन्स के अंदर एसिटाइलेशन एंड मिथाइलेशन एट द lysine is the most common. Ab, in what conditions are these modifications taking place and what are these modifications helping us to do? अगर histone molecule which is bound to DNA gets acetylated, then it leads to activation of transcription. एसिटाइलेशन से क्या होगा कि हिस्टोन मॉलिक्यूल्स का डीएनए के साथ बाइंडिंग थोड़ी कम हो जाएगी. दे विल जस्ट सेपरेट देमसेल्व्स फ्रॉम डीएनए एंड इट विल गिव डीएनए स्पेस एंड टाइम एंड place to do the transcription. उसके जो ट्रांसक्रिप्शन के एंजाइम्स है वो आके उसको बाइंड कर जाएंगे फटाफट डीएनए को, ट्रांसक्रिप्शन होनी शुरू हो जाएगी. ठीक है. सो एसिटाइलेशन इज हेल्पिंग इन द ट्रांसक्रिप्शन. Deacetylation, it will repress, do the opposite thing. It will become more tightly bound. It will cause de-repression of transcription. Phosphorylation will lead to condensation of the chromosomes, क्योंकि जितना phosphate होगा उतना और ज़्यादा वो condense हो जाएगा. ADP ribosylation is required at the time of DNA repair. Methylation is required to repress the gene. तो अगर methylation हो जाएगी तो genes जो है वो transcribe नहीं कर पाएंगे. They will remain in repressed stage. And simulation of histones causes again repression of transcription. So this is how the modification of this thing histones is affecting the DNA at all levels. So what are the functions of histone molecules? First is to condense the DNA and to make the molecule more compact. It is 40 times more shorter than unpacked molecule. And also to participate in gene regulation by undergoing different covalent modifications. And conserved packing material of DNA is important in chromosome stabilization and gene expression. Any doubts till now? So all is going fine, everybody is understanding? Okay, then I'll move further. Now we come to proper DNA organization. What does DNA organization mean? DNA organization में क्या हो रहा है कि eukaryotic organization has 4 main stages. First is nucleosome formation, then is 10 nanometer fibril formation, then is 30 nanometer fibril formation, and then comes the loops and domain formations. Nucleosome क्या है? So nucleosome formation is the first stage of DNA packing, and at this stage the DNA shows a specific type of look, that is the beads on string arrangement. Okay, there is a string which has beads on it, and these beads are the nucleosomes. And what is a nucleosome? Nucleosome on the electron microscopic view, chromatin which has condensed it into a dense spherical particle is called as a nucleosome. It has approximately a diameter of 10 nanometer and it is connected by DNA filaments in between. Nucleosomes are mainly composed of DNA which is wound around a collection of histone molecules. ठीक है. So now अभी तक जितना भी कुछ मैंने बताया उस सबको आप इकट्ठा जोड़ के देखें. So nucleosomes are beads on string appearance जो दिखता है इलेक्ट्रॉन माइक्रोस्कोप में, जिसका डायमीटर एक spherical molecule with a diameter of 10 nanometer with attached linker DNA in between, and वो octamer of histones के around DNA thread is wound, and this is a nucleosome. Then spherical particle of chromatin is the nucleosome. Nucleosome is formed of a histone core of 8 molecules of histones H2A, H2B, H3, H4 in pairs, around which segment of DNA double helix is wrapped nearly twice, left-handed superhelical turn, and organization of DNA double strand in nucleosome makes it to look like beads in a string-like look, and they lead transcription compaction of DNA up to 7 times. ठीक है. So this is how it looks. Core histone molecule, uske around DNA becomes a nucleosome. These nucleosomes are attached with the help of center ka jo beech ka linker DNA hai. And this is you can see the image where it is on the electron microscope. It looks like this beads on string appearance. और बीच में ये histone 1 in this नीचे वाला figure you can see histone 1 is stabilizing the molecule. It is attached from the outside. It's not in the core of the histone. So this is the first level of packaging. ठीक है? So again in this figure we can see 8 histone core, DNA आपका H1 वो और एक ये linker DNA. DNA is wrapped around the surface of a flat protein cylinder consisting of the core histone molecules, octamer, 146 base pairs of DNA consisting of 1.75 superhelix turns are in contact with the histone octamer. This protects the DNA from digestion by a nucleus. तो हमें एक तो हमें DNA को कॉम्पैक्ट करना है, उसको थोड़ा सा स्पेस में फिट करना है. दूसरा, इस तरह से कॉम्पैक्ट होने से DNA is protected from the digestion by nucleases. H1 is the most common, most tissue specific. It facilitates the packing of nucleosomes into more compact structures. Neighboring nucleosomes are joined by a linker stretch of DNA, which is approximately 50 base pairs and is covered by one molecule of H1, thus completing 2 DNA turns. And another thing which you should remember, that histone molecule is not bound randomly to the DNA, but at specific locations, which will have more of 80 regions. ठीक है, this by 80 region पे करना है क्योंकि this is less rigid than GC. GC में 3 bonds हैं, 80 में 2 bonds हैं, so this is less rigid. So 80 region जो होता है adenine और thymine pair, जहाँ पे ज्यादा जिस एरिया में होंगे वहाँ पे histone molecules attach होंगे. And this helps in the tight wrapping of DNA around the nucleosome core. And formation of nucleosome leads to compaction of DNA by approximately 7-fold. I have already told you. And after this packaging successfully goes to higher orders. Now, next order is the formation of 10 nanometer fibrils. Now, just like nucleosome structure, अभी तक बन गया. It has 10 nanometer diameter, वो भी मैंने बताया, and it has a height of 5 nanometers. This 10 nanometer fibril consists of nucleosomes which are arranged with their edges which separated by small distance, that is 30 base pair of DNA with their flat faces parallel to fibril axis. The diameter of each fibril is 10 nanometer, which you should always remember. And again, 7 times ज़्यादा इसका packaging से packing ratio है. Now we come to 30 nanometer chromatin fiber. तो अभी तक हमने समझ लिया, अभी तक हम nucleosome बनाया, 10 nanometer उसकी diameter है. अब 30 nanometer chromatin fiber क्या है? जो 10 nanometer fibrils बने हैं, they are further going to be organized into a higher order structure, which we call as 30 nanometer chromatin fiber. This 10 nanometer fibril is supercoiled into a circular 30 nanometer fiber with 6 nucleosomes per turn. You can see in this figure, there will be, I'll tell you the theory and then we'll come to the figure. So, 6 or 7 nucleosomes per turn to form 30 nanometer chromatin fiber. Each turn of supercoil is relatively flat, and the faces of the nucleosomes are successive turns would be nearly parallel to each other. H1 histones appear to stabilize the 30 nanometer fiber, but their position and that of the variable length spacer DNA is not very clear. And nucleosomes, they form back structure, and this 30 nanometer fiber must be compacted in the length order of 100-fold. Okay, in this you can see 30 nanometer fibril composed of, इसमें you can see there are 6 nucleosomes in one turn. Okay, and if we see this is a DNA. A part of DNA, when you see, it is arranged like nucleosomes, and this is the linker DNA, and this is the abrigo, and then this forms similar 6 such molecules will form 30 nanometer chromatin fibril. Again, I'm telling you, this 10 nanometer fibril is getting super coiled to form circular 30 nanometer fiber with 6 or 7 nucleosomes per turn. Each turn of this supercoil is flat with and faces of the nucleosomes of successive turn are parallel to each other, and histone 1 stabilizes this structure. Now we come to the loops and domains. In the interface chromosomes, chromatin fibers appear to be organized into 30,000 to 1 lakh base pair loops or domains, which are anchored in a scaffolding, that is a supporting matrix within the nucleus. Okay. Within these domains, some DNA sequences will be just located non-randomly. It has been suggested that each looped domain of chromatin corresponds to one or more separate genetic function, which that means it contains both coding and non-coding regions of the genes. And this nuclear architecture is very dynamic, and it has certain genes which are mobile within the nucleus, which move to discrete locus within the nucleus upon activation. So I will repeat all this again. Further compaction, which it leads to higher level of organization. Beyond 30 nanometer, we come to loops and domains. न्यूक्लियर स्कैफोल्ड रहता है, ठीक है, उसके ऊपर this region of DNA forms loops or domains which contain a set of functionally related genes. तो एक पर्टिकुलर जीन्स जो है वो लूप्स या डोमेन्स बनाते हैं, ठीक है. This scaffold is made up of several proteins, and this is around a scaffold, that is a protein molecule, and around that DNA will form a domain or looped domain, which will be particular genes का एक particular domain बनता जाएगा. Details इसकी बहुत ज़्यादा इतनी clear नहीं है, लेकिन you have to understand that it involves formation and compaction of coils upon coils. Coils के ऊपर coils बनती जाएंगी, ultimately leading to thick compact bunch of loops attached to a scaffold. ठीक है? Again, we will revise this so that you are able to understand it better. Nucleosomes are packed tightly to form polynucleosome, 6 to 7 nucleosomes in one turn to form 30 nanometer fiber. ठीक है, एक तो ये बन गया. So it looks like a solenoid, that is a cylindrical coil. ठीक है, you can see this एक ये चीज इसने एक दूसरे के ऊपर वो करके एक सिलेंड्रिकल लुक दे दिया, जैसे 30 नैनोमीटर का बन गया. ठीक है, वो टाइटली पैक हुआ. अब उसके बाद इसका क्या हुआ, the 30 nanometer fiber is organized into loop that are anchored to that nucleus scaffold containing several proteins. ठीक है, to give 700 nanometer structure. Again you can see ये आपका न्यूक्लियोसोम ने 30 नैनोमीटर फाइबर बनाया, इस तरह से 10 नैनोमीटर के न्यूक्लियोसोम से वो फोल्ड होकर के 30 नैनोमीटर के फाइबर बने. फिर एक प्रोटीन्स के फोल्ड के ऊपर इन्होंने लूप के ऊपर लूप बनाना शुरू कर दिया इस तरह से. ठीक है, और ये लूप्स आपस में कंडेंस होकर के क्रोमोसोम में चले गए. Are you able to understand now? I'm repeating it again and again, and I've tried to show different figures. Probably by this figure it will be more clear. You can see the protein scaffold, which is a nuclear scaffold which has proteins on it. Around it, this 30 nanometer wala jo solenoid bana tha, that will organize into loops. Okay? And then higher level of organization will be to form a chromosome, that all will get compacted and form a chromosome. So finally, I will revise this again. DNA molecule. This is the first is the DNA molecule that binds with histones, forming a vast nucleosomes. Then nucleosomes look like bead on string. These nucleosomes pack into 30 nanometer, 10 nanometer, and then 30 nanometer, and they produce condensed so-called 5 chromatin fibers. Then they attach on a protein scaffold to form loops, and then those loops even further they condense to form chromosomes. So now we have done this. Now I will talk about active and inactive chromatin. Chromatin containing active genes, which was euchromatin. So having active genes which are transcriptionally and potentially ट्रांसक्रिप्शनरी या आगे एक्टिव हो सकते हैं, किसी एक टाइम पे दे माइट नॉट बी एक्टिव बट आगे वो एक्टिव हो सकते हैं. सो दे आर डिफरेंट फ्रॉम दोज दैट आर ऑलवेज इनएक्टिव. कुछ तो ऐसे होते हैं जो मतलब जंक डीएनए जो कि ट्रांसक्रिप्शन में जाएगा ही नहीं. ठीक है. सो दे आर डिफरेंट. डीएनए in the active chromatin contains large regions which are sensitive to digestion by DNAs. These large regions, they इनके बीच में there are stretches of 100 to 300 nucleotides that exhibit even greater sensitivity to DNAs. These are hypersensitive sites, and they are all often located upstream, मतलब above से पहले from the active gene. ठीक है. Transcriptionally inactive chromatin जो होता है वो densely packed है during the interphase, and it is called as heterochromatin, and उसका stain भी less. active chromatin तो lightly stain होता है ना, heterochromatin जो है वो densely stain होता है. The chromatin in the heterochromatin region is high in the methylated cytosine. जो ये मैंने बताया था जो हिस्टोन्स में एस्टाइलेशन, डीएस्टाइलेशन और मिथाइलेशन होता है, ये तो हिस्टोन्स का हो गया. एक होता है कि जो आपका डीएनए है, डीएनए के जो जीन्स हैं या जो डीएनए के सीक्वेंसेस हैं उसके अंदर जो साइटोसिन्स हैं, नॉर्मली दे आर मिथाइलेटेड सो दैट जब डीएनए ने ट्रांसक्रिप्शन नहीं करनी, आरएनए नहीं बनाना, उस समय उसको कोई सिग्नल तो चाहिए कि मुझे इनएक्टिव रहना हर समय, अदरवाइज वो प्रोटीन बनाता रहेगा, आरएनए बनाता रहेगा. हर समय तो आपको नहीं चाहिए. जिस समय आपको चाहिए उस समय आप वो मिथाइलेशन साइट्स पे एक्सपोज करते हो. अदरवाइज जो आपका क्रोमैटिन के अंदर रीजन्स ऑफ इनएक्टिविटी, जब आपको ट्रांसक्रिप्शन नहीं चाहिए उस समय जो आपके साइटोसिन्स हैं दे आर मिथाइलेटेड. ठीक है. दे आर हाई इन मिथाइलेटेड साइटोसिन कंटेंट, and the histones therein contain relatively lower levels of covalent modifications. हमने देखा था कि covalent modifications जो है वो repression कर रही थी, उसको depress कर रही थी. So low levels of covalent modifications of the histones are there in the inactive chromatin area. So this we have already done. एक आपका मैंने क्रोमैटिन बताया, यूक्रोमैटिन एंड हेट्रोक्रोमैटिन. एक है कॉन्स्टिट्यूटिव एंड फैकल्टेटिव. देयर आर टू टाइप्स ऑफ हेट्रोक्रोमैटिन, कॉन्स्टिट्यूटिव एंड फैकल्टेटिव. कॉन्स्टिट्यूटिव मींस which is always condensed, thus essentially inactive. It is found in the regions near the chromosomal centromere and at the ends, that is centromere और telomere के पास. Facultative is the one which is at times condensed, but other times it is actively transcribed, जिसकी ट्रांसक्रिप्शन होनी है, जिससे आरएनए बनना है, वो facultative है. Thus, they are uncondensed in those areas and appear as euchromatin. two members of the X chromosome pair in the mammalian females, one X chromosome is almost completely transcriptionally inactive. That is the heterochromatic. However, the heterochromatic X chromosome decondenses during gametogenesis and becomes transcriptionally active during embryogenesis, thus becomes facultative heterochromatin. Okay? So this you have to, if you understand this mechanism, that the heterochromatin which changes from inactive to active state in the cell is the facultative heterochromatin, and sex chromosome is usually heterochromatic but becomes euchromatic at the time of gametogenesis. Another term which I wanted to introduce is the polytene chromosomes. Like in some insects, Chiromonomus and Drosophila, they contain giant, huge chromosomes that have been replicated for multiple cycles without separation of the daughter chromatids. ठीक है, मल्टीप्लाई होता जा रहा है लेकिन डॉटर क्रोमैटिड सेपरेट नहीं हो रहे. अदरवाइज यूजुअली तो माइटोसिस में ऐसे ही होता है कि डॉटर क्रोमैटिड सेपरेट होते जाते हैं, न्यूक्लियर सेल बनते जाते हैं. एक ही सेल में अगर डॉटर क्रोमैटिड सेपरेट ना हो लेकिन मल्टीप्लिकेशन होती जाए तो देयर क्रोमोसोम विल बिकम जायंट. ठीक है, सो दीज बैंडेड क्रोमोसोम्स कंटेन रीजन्स ऑफ द condensed chromatin and lighter bands of more extended chromatin. These are the polytene chromosomes. How does this chromatin remodeling take place and what is chromatin remodeling? The process of structural changes occurring in the heterochromatin to become euchromatin is the chromatin remodeling. जो heterochromatin है आपका, that is transcriptionally inactive, वो जब जब euchromatin में convert होगा जो कि ट्रांसक्रिप्शनली एक्टिव है, तो उस प्रोसेस को हम बोलेंगे रीमॉडलिंग. एंजाइमेटिक मैकेनिज्म्स to modify histone proteins and it uses ATP. There is acetylation and deacetylation. Amino terminal domains of the core histones are rich in arginine and lysine. And there is this hat enzyme, cytosolic hat enzymes होते हैं. ठीक है, histone acetyl transferases. The main mechanism includes the acetylation and deacetylation of the core histone molecules through histone acetylation transferases. ठीक है, so these enzymes acetylate the core histone molecules which are rich in arginine and lysine, and they reduce the affinity of nucleosome for DNA, which promotes the binding of transcription factors. I told you this. When अगर आप हिस्टोन्स को एसिटाइलेट कर दोगे तो उसके अंदर बाइंडिंग ऑफ हिस्टोन्स टू न्यूक्लियोसोम्स कम हो जाएगी एंड वो उसको न्यूक्लियोसोम्स को ट्रांसक्रिप्शन फैक्टर्स के साथ बाइंडिंग का एफिनिटी बढ़ जाएगी. हिस्टोन्स को रिलीज करके they will bind to the transcription factors and become transcriptionally active. When transcription is not required, they act to another thing. हमें जीन कल ही इंट्रोड्यूस कर दिया था वैसे तो, लेकिन exactly what is a gene? How do you define a gene? Gene is the part of DNA, the DNA stretch, which encodes for a specific protein. तो पूरा DNA को हम जीन नहीं कहते, लेकिन जीन DNA का वो पार्ट जिससे एक प्रोटीन बनना है, उसकी सीक्वेंस उसमें दी हुई है, उस पार्ट ऑफ डीएनए को, उस सीक्वेंस ऑफ डीएनए को हम जीन बोलते हैं. DNA is a big biomolecule which has the genetic information and it regulates the traits of all the organisms. Gene is the specific sequence present on short stretch of DNA. So अभी तक सब क्लियर चल रहा है? Anybody can write in the chat box. So everything is clear till now? Hat enzymes are histone acetylase transferase. I told you कि histones जो है उनके अंदर कोवेलेंट मॉडिफिकेशंस हो रही हैं, एसिटाइलेशन, डीएसिटाइलेशन. तो एसिटाइलेशन कैसे होगी? उसके लिए जो एंजाइम एसिटाइलेट ग्रुप को ऐड करेगा हिस्टोन्स में, उसको कहते हैं हैट हिस्टोन एसिटाइल ट्रांसफरेज एंजाइम. They modify, chemically modify, covalently modify the histone molecules so that their binding to the DNA becomes less, it has less affinity, so that the DNA की जो properties हैं वो change हो सके. Now I move further. Gene and genome. Gene is the fundamental unit of heredity. It is a segment of DNA on a specific position. अब इस specific position को हम locus भी कहते हैं, ठीक है? So segment of the DNA on a specific position, that is called the locus on the chromosome. Each gene is a part of DNA sequence that contains genetic information coding for synthesis of one polypeptide protein. Genome is the total number of genes within one mature cell of the organism. Genome तो whole genome, सारा का सारा DNA, सारा का सारा genes, उसको हम genome कहते हैं. लेकिन just the part of that stretch of DNA जिससे एक प्रोटीन, जिसके अंदर उस RNA की सीक्वेंस है, क्योंकि RNA सारे DNA को नहीं बना रहा है ना, DNA के अंदर तो बहुत सारे प्रोटीन्स की सीक्वेंसेस है. तो एक छोटा सा पार्ट ऑफ DNA एक छोटा सा RNA बनाएगा, वो छोटा सा RNA एक प्रोटीन बनाएगा. तो एक प्रोटीन का इंफॉर्मेशन जिस पार्ट ऑफ DNA से आई है, उस पार्ट को हम जीन कहते हैं, ठीक है, लोकस. Only 10% of the human DNA contains proper genes, and the remaining 10% of the DNA is important as regulatory and identify functions. जो सीक्वेंसेस जिनके अंदर जीन बनाने की कैपेसिटी है, वो सिर्फ बहुत थोड़े से 10%. बाकी तो सारे उसके रेगुलेटरी सीक्वेंसेस है. कहाँ पे कोई ट्रांसक्रिप्शन फैक्टर बाइंड करेगा, कहाँ पे कोई रेगुलेटरी फैक्टर प्रोटीन बाइंड करेगा, तो वो सीक्वेंसेस अलग है. They are not called genes. Another fun factor, our genes are remarkably similar to those of the other life forms. Like we share 98% of our gene with chimpanzees, 90% with mice, 85% with zebrafish, 21% with worms, and 7% with a simple bacterium such as E. coli. What are the different types of genes? The housekeeping genes हैं और differential specific function के genes हैं. Housekeeping genes are the genes that are essential for life, and they are integral parts of the cells, like hexokinase gene that is essential for glucose metabolism inside every cell. अब glucose तो basic आपका जो भी खाना है ग्लूकोज बन रहा है और उसका मेटाबॉलिज्म होना, उसका जीन तो हर सभी सेल को चाहिए. So they are housekeeping genes essential for life, which are active all time. Differential specific function of genes, these are the genes that are essential for performance of specific functions, and so they differ in each organ. Genes which are specific for liver function will be expressed only in liver cells. इंसुलिन वाले सिर्फ पैंक्रियास में होंगे. तो ये स्पेसिफिक फंक्शन वाले डिफरेंशियल जीन्स है. Structure of gene. Size of each gene is variable, depends upon the number of amino acids of the proteins it codes. Okay? They can have intervening non-coding sequences and other regulatory sequences. Prokaryotic genes will have no. अच्छा, prokaryotic genes में जो क्या होता है, पहले eukaryotic समझ लो. They have non-coding regions which are called as introns intervening in between the coding segments, which are called the exons, which are expressed. Exons मतलब express हो रहे हैं, जो RNA बना रहे हैं, उनको हम coding segments कह रहे हैं. उनके बीच में DNA के अंदर non coding sequences हैं जिनको हम intron कह रहे हैं, क्योंकि वो intervene कर रहे हैं exons के बीच में. जब मैच्योर मैसेंजर RNA बन जाता है, तो उसके अंदर मैंने कल भी बताया था कि इंट्रॉन्स जो है वो रिमूव हो जाते हैं और एक्सॉन्स जुड़ जाते हैं और एक मैसेंजर RNA मैच्योर बच जाता है. लेकिन प्रोकैरियोट्स के अंदर ऐसी कोई इंट्रॉन्स नहीं होते, कोई इंटरवीनिंग नॉन कोडिंग सीक्वेंसेस नहीं होती, सिर्फ रेगुलेटरी और कॉड कोडिंग सीक्वेंसेस होती हैं. बैक्टीरिया हैव वन क्रोमोसोम विद वन कॉपी ऑफ जीन. How big can be a gene? A polypeptide chain of 350 amino acids अगर है, तो it will correspond to 3 times, इसके 3 times एक amino acid 3 न्यूक्लियोटाइड से आ रहा है ना, तो 350 into 3 will be 1050 base pairs coding DNA. So genes are interspersed between the नॉन कोडिंग सेगमेंट्स. तो इक्वलाई के अंदर आप देख लो ये 46 lakhs base pairs will encode 4300 genes for proteins and 157 genes for structural and catalytic RNA molecules. तो base pairs देखो कितने ज्यादा है. Now we come to introns. We will see some more facts about it. Gene and chromosome. एक क्रोमोसोम को अगर आप थोड़ा सा पार्ट उसका देखें तो उसके अंदर यू कैन सी ये डीएनए है ना, तो इसके अंदर यू कैन सी जो डार्क बैंड्स हैं वो तो आपके हो गए एक्सॉन्स जो एक्सप्रेस होने हैं. उसके अंदर ये अराउंड 20 जीन्स उसने दिखाए हैं जो इंटरवीनिंग लाइट वाले बैंड्स जो हैं ब्रॉड ब्रॉड से वो इंट्रॉन्स हैं. तो उसको अगर हम और पास से देखें यू कैन सी introns may serve to separate functional domains, that is the exons of coding information in form that permits genetic rearrangement by the recombination of the molecule to occur more rapidly than if the coding regions of the given genetic function were contiguous. Such an enhanced rate of genetic rearrangement of functional domains might allow more rapid evolution of biological function. Now I will introduce another term, jumping genes or transposons. These are the transposable elements called as jumping genes. They are the sequences of DNA that move or jump from one location to in the genome to another. अब jump ऐसे नहीं होता है. Almost half of our DNA sequences are made up of jumping genes. That is, they can move from one particular position to another. What happens in bacteria? There are two types sequences. एक होती है simple sequences, एक complex sequences that are to be moved along with the gene of for transposase. इसमें कोई और जीन नहीं होता है, सिर्फ एक सीक्वेंसेस होती हैं जो ट्रांसपोजेज एंजाइम होता है उसके साथ मूव करती हैं. कॉम्प्लेक्स में एडिशनल जीन्स व्हिच गिव एंटीबायोटिक रेजिस्टेंस दे आर आल्सो इनकॉर्पोरेटेड. मैकेनिज्म ऑफ ट्रांसपोजिशन दैट इज जंपिंग जीन कैसे वो कैसे रहा है. एक प्रॉपर्टी आ जाती है फॉर डेवलपिंग द एंटीबायोटिक रेजिस्टेंस. So again I will tell you what is the simple and complex transposon. बैक्टीरिया के अंदर दो टाइप के ट्रांसपोसॉन्स होते हैं, सिंपल एंड कॉम्प्लेक्स. सिंपल इज द सीक्वेंस दैट can move along with the gene sequence that codes for the enzyme called transposase. audible now? Okay. So, from which slide you want me to repeat? So, transposons, I have told these are the jumping genes, and these are the jumping genes, and am I audible now? Okay, 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 got it, got it. So let us continue से पहले कि कुछ और प्रॉब्लम हो सिस्टम में. So the human genome, I told you the characteristics. इससे ज़्यादा कुछ समझाने की बात नहीं है. It is just to understand. Haploid genome has 3 into 10 to the power 9 base pairs of sequences divided into 23 chromosomes, and haploid genome has sufficient DNA to code for 1.5 million average size genes. And जो इसका purpose binding to give the binding sites for regulatory proteins to encode for regulatory RNAs, microRNAs like things. It is made up of introns, ठीक है. It is composed of many families of repeated sequences. अब ये repeated sequences की बात हम करेंगे. एक मैंने junk DNA के बारे में बताया था कि junk DNA is the part of the DNA which is non-protein coding. It is never translated into a protein molecule. ठीक है. So exons जैसे हमने पहले डिस्कस किया था कि they are the ones which are coding sequences sections of the RNA transcript, or introns are the non-coding sections. So junk DNA is the non-coding part which is never translated, and it is इसमें एक चीज और याद रखनी है, वेरी इंपॉर्टेंट थिंग, कि डीएनए का जो कोडिंग पार्ट है, इट फॉर्म्स, व्हाट डज इट फॉर्म? इट फॉर्म्स मैसेंजर आरएनए. ठीक है. लेकिन जो बाकी टाइप्स ऑफ आरएनए है, जैसे आर आरएनए, टी आरएनए, माइक्रो आरएनए, एसए आरएनए, ये सारे आरएनए कहाँ से बन रहे हैं? ये नॉन कोडिंग, सो कॉल्ड नॉन कोडिंग एरिया से बन रहे हैं. ठीक है. सो नॉन कोडिंग एरिया का ये मतलब नहीं कि वो बिल्कुल ही जरूरी नहीं है. तो डीएनए कंटेन्स इंस्ट्रक्शंस दैट आर यूज्ड टू क्रिएट प्रोटीन्स, एंड सम ऑफ दैट इज, वो तो हो गया आपका मैसेंजर आरएनए कोडिंग. लेकिन the non-coding DNA is used to produce non-coding RNA components like tRNA, regulatory RNA, and ribosomal RNA. Junk DNA is the non-coding region which is giving other important functions. Other examples of the junk DNA are satellite DNA, telomeres, introns, non-coding RNA genes. जो RNA encode कर रही हैं genes, चाहे वो tRNA है चाहे rRNA, वो हम junk में नहीं include करेंगे. ठीक है. And the DNA regulatory sequences. एक और चीज होती है रिपीटेटिव एंड नॉन रिपीटेटिव सीक्वेंसिंग. सीक्वेंस जो है आपकी डीएनए की, जो सीक्वेंस है बेस पेयर्स की, वो एक स्पेसिफिक फॉर्मेट में है. तो एक तो होती है नॉन रिपीटेटिव डीएनए सीक्वेंसेस, एक है रिपीटेटिव डीएनए सीक्वेंसेस. नॉन रिपीटेटिव डीएनए सीक्वेंसेस जो हैं, दे आर मोर देन हाफ द डीएनए इन यूकैरियोटिक ऑर्गेनिज्म इज इन यूनिक और नॉन रिपीटेटिव सीक्वेंस, दैट इज कोडिंग समथिंग. ठीक है. सम ऑफ इट विल बी कोडेड, mRNA बनेगा और प्रोटीन बनेगा. Some of them will make other things like it will have introns, regulatory genes, pseudo genes, etc. But that sequence will be AG, CAG, some random, non-repetitive. Some will be repetitive sequence DNAs, that is 30% of the genome consists of repetitive sequences. They are classified as moderate. Am I audible now? I don't know what is happening today. So highly repetitive sequences consist of 500 to around 5 to 500 base pairs which will be repeated in tandem. अगर मान लीजिए 10 base pairs का कोई आप sequence ले लो, GCA, GCT, अब वही repeat होता जाएगा बार बार 500 times, कितना times. This is clustered in centromeres or telomeres of the chromosomes, and it is present about 1 to 10 million copies per haploid genome. Transcriptionally, it is inactive. Some of these sequences play a structural role in the chromosome. Moderately repetitive sequences जो हैं, they can be long interspersed repeat sequences or short interspersed repeat sequences, lines or signs. ठीक है. तो जो ये long interspersed है, they consist of around 6,000 to 7,000 base pairs, long sequences which will be present in 20,000 to 50,000 copies in genome. ठीक है. मतलब इतने लंबे सीक्वेंसेस हैं और वो आपके कितने टाइम्स 20,000 50,000 टाइम कॉपीज इसकी प्रेजेंट हो सकती हैं लॉन्ग इंटरस्पर्स्ड रिपीट सीक्वेंसेस. ठीक है. और शॉर्ट जो है वो 70 to 300 बेस पेयर्स की सीक्वेंस होगी which will be present, it can be repeated around 1 lakh times in the genome. अब इसमें इम्पोर्टेंट चीज है साइन जो है, इसके अंदर एक फैमिली है एलू फैमिली. एलू हमने नाम इसलिए दिया है क्योंकि इसके अंदर एक रिस्ट्रिक्शन एंडोन्यूक्लियस एंजाइम होता है LU1. LU1 is the restriction endonuclease enzyme which cuts the DNA from inside. ठीक है, रिस्ट्रिक्शन साइट बोलते हैं उसको. तो LU family का जो एक सीक्वेंस है, it consists of moderately repeating sign sequence about 300 base pairs which may be present up to 5 lakh copies in the genome. They are highly conserved, so they are called LU1 because it has the restriction site for LU1 restriction endonuclease. It can lead to neurofibromatosis. Now, I think this must be the last thing, satellite and microsatellite repeat sequences. Satellite DNA is a portion of the DNA which consists of tandem repeats of non-coding regions. अभी फिर से ऑडिबल हो गया ना? Abhi it's audible? I'm just about to finish the class now. Satellite DNA is a portion of the DNA that consists of tandem repeats of non-coding nucleotide sequences that occur in the genome. Tandem repeats मतलब जो बार-बार repeat हो रहा है. They are the copies of nucleotide sequences that lie adjacent to each other. The length of a satellite DNA ranges from 1 to several base pairs. 2 to 6 base pair repeated up to 50 times को हम tandem repeat बोलते हैं. Microsatellite sequences, they are commonly found as dinucleotide repeats of like AC on one strand and TG on other opposite strand. But several other forms also occur like CG, AT, CA. ठीक है, इस तरह से कोई भी forms की tandem repeats को हम satellite बोलते हैं, satellite repeats, microsatellite repeats. AC repeat sequences occur at 50,000 to 1 lakh locations in the genome. So the number of microsatellite repeats at a given location on two homologous chromosomes is usually different. Usually this microsatellite thing microsatellite DNA is associated with most genes and it serves as a marker for them. ठीक है, तो हम किसी relative position of gene को identify करने के लिए microsatellite genes को use करते हैं as markers, क्योंकि हमें पता है इस gene पे इतने tandem repeats हैं. अभी तो human genome पूरा identify हो चुका है, so you can use the microsatellites and the satellite sequences as gene markers. Now we come to this is the last slide, epigenetics and epigenome. Genome is the genetics that is the complete genetic information. Either DNA or some viruses में RNA हो सकता है, RNA viruses भी हो सकते हैं, DNA viruses भी हो सकते हैं. The complete genetic information of the organism is the genome of that organism. Epigenome मतलब the chemical which is a chemical reaction or a chemical difference responsible for the activation of a particular gene. Like multitude of chemical compounds that can tell the genome what to do. Like हमने क्या methylated होते हैं कुछ cytosines genes के अंदर so that the gene वो gene जो है वो transcribe ना हो सके, है ना, उसको repressed रखने के लिए transcription कर दिया जाता है, उसको transcription रोकने के लिए methylate कर दिया जाता है cytosines को. So that is the chemical reaction compound, chemical group that has been added to that gene. So the chemical compounds that tell the genome what to do, chemical compounds and proteins that can attach to DNA and direct the actions such as turning the genes on is the epigenome. Epigenetics is the study of how your behavior or your environment can cause the changes that affect the way your genes work. Like DNA methylation, I told you methyl groups are added to the DNA molecule, especially the cytosine molecule, which prevents a certain genes from getting expressed. Also histone modifications, they also guide कि repression करना है, transcription को activate करना है कि deactivate करना है. इसके अलावा your other lifestyle modifications like diet, obesity, physical activity, smoking, alcohol, environmental pollutants, your working on night shifts, physical, any kind of stress, all these kind of things, they change the chemical composition of the DNA. Sequence of the DNA is same. Sequence means genome, but uh, when the sequence is when the sequence is same, but the chemical groups attached on those molecules are changed, then also it affects the transcription of your DNA. That is the epigenetics. Chemical composition which is activating a particular gene or inactivating a particular gene is the epigenetics. Okay, so I think this is end of my lectures. Want me to repeat anything or we can call it a day? I hope everything was clear. And I don't know why today this audio problem was happening, and I had to repeat something, or probably हो सकता है मैंने कुछ miss किया हो. But you are all always welcome. You can come to me anytime if you want to understand anything. So anybody wants to repeat as I stop. Okay, then I'm just ending the session. Okay, bye then everybody. Repeat what? Repeat the last slide. Genetics and epigenetics. Okay, I'll repeat. See, genetics means genome का genome means the sequence of the DNA. अब कोई वायरस के अंदर आरएनए भी हो सकता है, कोई आरएनए वायरसेस होते हैं, कोई डीएनए वायरस होते हैं. तो जो जेनेटिक इंफॉर्मेशन किसी ऑर्गेनिज्म की है, चाहे वो डीएनए फॉर्म में है, चाहे वो आरएनए फॉर्म में है, किस सीक्वेंस में वो आपका पैटर्न अरेंज्ड है, उसको हम कहते हैं जीनोम. अब इस जीनोम के अंदर मैंने आपको बताया कि कोवेलेंट मॉडिफिकेशन एट द लेवल ऑफ हिस्टोन्स हो सकती है, एसिटाइलेशन, डीएसिटाइलेशन, सिमुलेशन, मिथाइलेशन, है ना, वो सारी चीजें हो रही थी. वो सारी चीजें आपके आगे क्या कर रही थी, डीएनए का जो ट्रांसक्रिप्शन है, यानी कि आरएनए के अंदर फॉर्मेशन को कहीं पर रिप्रेस कर रही थी, कहीं पर एक्टिवेट कर रही थी. तो डीएनए की प्रॉपर्टीज को चेंज कर रही थी. सो दैट इज द एपिजेनेटिक्स. उसके अलावा डीएनए के अंदर जो साइटोसिन होता है, जिस लेवल पे, अगर हमें डीएनए को रेप्लिकेशन ट्रांसक्रिप्शन नहीं करना जिस समय, उस समय डीएनए के वो साइटोसिन मॉलिक्यूल्स मिथाइलेटेड होते हैं. ठीक है, और addition of methyl groups to the DNA prevents a certain genes from being expressed. ठीक है, कुछ एरियाज मिथाइलेट कर दिए जाते हैं ताकि वो जीन्स जो है वो एक्सप्रेस नहीं कर पाए. सिमिलरली आई टोल्ड यू हिस्टोन्स भी मॉडिफाई होते हैं, और कुछ आपकी लाइफस्टाइल मॉडिफिकेशंस जो है वो भी आपके हिस्टोन्स को या डीएनए मॉलिक्यूल्स को केमिकली मॉडिफाई करती है, कोवेलेंट मॉडिफिकेशन. सो दिस प्रॉपर्टी ऑफ मॉडिफिकेशन ऑफ द जीन ऑफ द डीएनए, जिसमें आपका सीक्वेंस ऑफ डीएनए चेंज नहीं हो रहा, ओनली केमिकल ग्रुप्स आर बीइंग एडेड और लेस एडेड टू द सीक्वेंस, इज कॉल्ड एज एपिजेनेटिक्स. जंपिंग जीन्स मैंने 3-4 बार बता दिया, इसमें भी कुछ खास बड़ी चीज नहीं है. जंपिंग जीन्स और स्यूडो जीन्स में बस इतना ही है कि जंपिंग जीन्स आर दी ट्रांसपोसॉन्स, इनको हम ट्रांसपोसॉन्स भी कहते हैं। दीज आर द सीक्वेंसेस ऑफ डीएनए दैट जंप फ्रॉम वन लोकेशन टू अनदर। इसमें क्या होता है, एक तो कर सकते हैं कि कुछ कुछ एक ट्रांसपोजेज एंजाइम होता है जो कि कुछ सीक्वेंसेस को कट करके अपने साथ इट कैरीज टू सम अदर पोजीशन। ठीक है, एक तो ये डायरेक्ट हो गया। दूसरा है कि जिस पोजीशन पर वो सीक्वेंस है उसी पोजीशन पे तो वो उसकी कॉपी वैसी रहती है, लेकिन न्यू पोजीशन पे जहाँ ट्रांसपोजेज जा रहा है वहां पे उसकी न्यू कॉपी रेप्लिकेट हो जाती है। ठीक है, तो ये हुए जंपिंग जीन्स। these are the retro trans और एक retro transposons भी होते हैं। इसमें almost half of our DNA sequences are made up of jumping genes. Pseudogenes होते हैं which are the process genes, जिसके अंदर डीएनए का सीक्वेंस इज सिमिलर सेम टू दैट ऑफ द आरएनए। ये नॉर्मली ऐसे नहीं बनता, लेकिन ड्यूरिंग इवोल्यूशन कहीं पर ऐसा हुआ होगा कि एक स्टॉप कोडॉन आ गया और प्रोसेस्ड आरएनए जो था उससे बैक रिवर्स कॉपी हो गई और डबल स्टैंडर्ड डीएनए बन गया। अब ये नॉन फंक्शनल डीएनए है विच इज रिजेंबलिंग द फंक्शनल जीन, बट दिस इज एक्चुअली नॉन फंक्शनल बिकॉज इसमें इंट्रॉन्स नहीं है। इसके अंदर ओनली एक्सॉन्स का सीक्वेंस है और पॉली ए टेल है, सेम एज द मैसेंजर आरएनए। दीज आर द स्यूडोजीन्स। ठीक है देन। Okay, then I'm just ending the session. Okay, then bye.